Hey, so we're getting close to the end of the class, you guys. Let's talk about oxidation of unsaturated fatty acids. Many of the fats in our diets, especially the oils, they're going to contain unsaturated fatty acids, which have one or more double bonds. So remember, I told you if it's saturated, we're absolutely saturated with all the hydrogens we could ever want, which means that we don't have room for any double bonds. Where when it's unsaturated, we have a double bond, and that's going to kick off at least 2 hydrogens per bond. It will exactly 2 hydrogens per bond. So since unsaturated fats are ready for hydration, no FADH2 is formed in this step. And then the energy from beta oxidation is slightly less. And so for simplicity, we're going to assume that the total ATP production is the same for saturated versus unsaturated fatty acids. So we have to talk about ketone bodies as well. Okay, so if carbohydrates are not available, sugars are not available, the body breaks down our fat stores to meet those energy needs. Because of this, ketone bodies form in a process that's called ketogenesis. Genesis meaning to make. In ketogenesis, acetyl-CoA molecules combine to produce ketone bodies, acetoacetate, and beta-hydroxybutyrate, and acetone. All right, so basically think of the keto diet. What do? You cut out sugars. What do you break down and stuff then? Well, fats. And so this would be kind of how you can equate the ketone bodies. So ketone bodies form if large amounts of acetyl-CoA accumulate, or when 2 acetyl-CoA molecules form acetoacetyl-CoA, or when acetyl-acetyl-CoA hydrolyzes to acetoacetate, or when acetoacetate reduces beta-hydroxybutyrate or loses the CO2 to form acetone, and these are both ketone bodies. So what is ketosis? Ketosis occurs badly in diabetics or diets that are high in fat, and actually starvation as well. And as ketone bodies accumulate, then the acidic ketone bodies are going to lower the blood pH below 7.4 because ketones are acidic. Okay, so in diabetics it's bad. Sometimes when you're purposely doing it for short periods of time, like the keto diet, it's okay. So how do we make fats from carbohydrates then for storage? So when the body has met all of its energy needs and the glycogen stores are full, then extra acetyl-CoA from the breakdown of carbs or sugars are going to be used to form new fats. So this would be like your lovely love handle that will never go away. In this process, 2 carbon acetyl units are linked to give a new fatty acid, and several reactions in the synthesis or making of fatty acids are the reverse of the oxidation reactions of fatty acids. And then the synthesis or making occurs in the cytosol instead of in that mitochondria. Then the new fatty acids are going to attach to glycerol to make triglycerides or triglycerols that we talked about before to be stored as body fats. So let's link this to health. The U.S. Public Health Service now estimates that in the United States, more than 1/3 of adults are obese. And obesity is where you have a body weight that's more than 20% over your ideal weight, and it's a major factor in health problems such as diabetes, heart disease, high blood pressure, stroke, and some cancers. And new research indicates that certain pathways in lipid and carbohydrate metabolism may be a possible cause for excessive weight gain. Now, I'm reading a book right now, and it's kind of interesting, but it's all about weight and weight loss. It's called The Obesity Code. It's actually pretty interesting because it talks about the actual science behind like your basal metabolic rate, etc. But they talk a lot about leptin. And so this is a relatively newer discovery where in 1995, scientists discovered that a hormone called leptin is produced in fat cells. And when fat cells are full, high levels of leptin signal the brain to limit the intake of food. It's like your I'm full hormone, right? And so when fat stores are low, then the leptin production decreases. Then this tells your brain, hey, you should probably eat again, And then it acts on the liver and the skeletal muscles where it stimulates fatty acid oxidation in the mitochondria, and therefore breaking it down is going to decrease fat storage. So if you want to learn more about leptin and obesity, scientists are studying the differences in the rate of leptin production, the degrees of resistance to leptin, and possibly the combination of these factors. Like I said, I'm reading a really cool book if you want to know about it. So great, that's about fats. Well, we also have proteins that our body breaks down. So how do we break down or degrade amino acids? Well, once the carbon skeletons of the amino acids are obtained from transamination, then they can be used to become intermediates of the citric acid cycle and therefore provide reduced coenzymes for the electron transport chain. So we're going to do this and learn how to describe the reactions of transamination, oxidation, oxidative deamination, and the entry of amino acids and carbons into the citric acid cycle. So proteins, they provide energy when carbs and lipid resources are not available. So your body, like I said last week, it ranks things in preference. The first thing your body wants to break down is sugars. The seconds would be fats, and the third would be proteins. So that's why if you're like thinking of, okay, you're deserted on a desert island, unfortunately, first thing you're going to burn through is going to be your sugars. Second, you're going to have fats, right? So then you're going to have zero body fat content. And then the third would be your actual protein or your muscles, okay? And then that's really bad when you start breaking down your own muscle for nutrition. And so the carbon atoms from the amino acids are used in the citric acid cycle for the synthesis or making of fatty acid, ketone bodies, and glucose. And most of the amino groups are converted into urea. All right, so what is this fun word, transamination? So in transamination, amino acids are degraded in the liver, and an amino group is transferred from an amino acid to an alpha keto acid, and this is usually alpha ketoglutarate. And a new amino acid and alpha keto acid are formed. And when alanine combines with alpha ketoglutarate, pyruvate and glutamate are produced. So this would be that reaction there, where you have alanine, which is one of our amino acids, alpha ketoglutarate, your alanine aminotransferase, and that's the enzyme that's actually doing the reaction. It's going to be converting it into pyruvate, which is that product of glycolysis, right, as well as glutamate. And so then this can go on and go into metabolism, etc. So in oxidative deamination, this is going to remove the ammonium or the NH3 group from glutamate as an ammonium ion, NH4+, and it's going to provide hydrogens for the NAD+ coenzymes. This is then going to act to regenerate the alpha ketoglutarate, and then this can enter into the transamination with an amino acid. So again, here's that pathway drawn out. You have glutamate, then you have your glutamate dehydrogenase, right? Dehydrogenase, I'm sorry. That's the enzyme that's doing the reaction, and you're going to be forming alpha ketoglutarate, etc. We then have the urea cycle. And the urea cycle is going to remove toxic ammonium ions from the amino acid degradation, and it converts those ammonium ions into urea in the liver. And this produces 25 to 30 grams of urea daily for excretion in the urine. So let's see how we're doing. Let's write the structures and the names of the products for the transamination of alpha ketoglutarate by aspartate. So pause me and come back. All right, so did you get this? Right, so we had that to start with, and then we're going to turn it into oxaloacetate and glutamate. So great job. All right, I'm just going to finish off this chapter instead of making another video. Okay, so ATP basically getting ATP from amino acids, which are our proteins. So this is the last step that we kind of don't really prefer to do because that means that we're in severe starvation. So carbon skeletons of amino acids form intermediates in the citric acid cycle. They produce energy and they enter the citric acid cycle at different phases or places depending on the amino acid. So if we have 3 carbon skeletons like alanine, serine, and cysteine, then it's going to enter at pyruvate. If we have 4 carbon skeletons like aspartate and asparagine, those are going to enter at the oxaloacetate level. Where if we have 5 carbon skeletons, we have glutamine, glutamate, and proline, as well as arginine and histidine, and these are going to enter at the glutamate level. So the intermediates then of the citric acid cycle from amino acids, you can see the alanine, cysteine, glycine, serine, and threonine are going to enter at the pyruvate. Where your isoleucine, the leucine and tryptophan are at acetyl-CoA. Leucine, lysine, and phenylalanine are at the acetyl-CoA. You have your ketogenesis that can produce the keto bodies, and then etc. All these different areas that we talked about. All right, so let's wrap up this class, you guys. Our last study checks. So once the carbon skeletons of amino acids are obtained from transamination, they can be used as intermediates of the citric acid cycle to provide reduced coenzymes for electron transport. So match each of the amino acids with the intermediate in the citric acid cycle. Pause me and come back. All right, cysteine is going to be pyruvate. Glutamine is alpha-ketoglutarate. Asparagine is fumarate, and serine is going to be pyruvate. So our overall view of metabolism then is going to be, in these metabolic pathways that we've seen, catabolic pathways degrade or break down large molecules into the smaller molecules that are used for energy production, where anabolic pathways are going to build up or lead to the synthesis of larger molecules in the cell. And there are several branch points in which the compounds can be degraded for energy or used for making or synthesizing larger molecules. So here is your overall view of metabolism. In the book, it's bigger, obviously, but you can see that we have our stage 1, stage 2, stage 3, and we end up making a whole bunch of ATP. And then here is your metabolic pathway concept maps of proteins, carbs versus fats or triglycerides. It's been a pleasure having you guys in class this semester, and good luck on your final. Thanks.